Okay, let's get started. I'm Eric Nestler. I'm director of the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai. I am an MD PhD myself, and it is a great pleasure to introduce you to today's event that features Dr. Dianna Milewicz, who will be talking to us about gender equity for physician scientists. This is a topic that I've seen firsthand in my efforts here at Mount Sinai to promote the recruitment of women to our MD PhD program. I know that this is a national challenge. While we now have a majority of women in medical school, a majority of women in PhD programs around the country, we still have not achieved that type of equity for MD PhD programs. Question is why, and what can we do to improve the situation? It is a great pleasure to introduce one of our own MD PhD students, a fourth-year student, Michelle Tran, who will introduce today's speaker. Michelle, take it away.
Thanks so much for those remarks, Dr. Nestler. On behalf of Sinai's Women in MSCP, I'm really pleased and excited to welcome Dr. Milewicz, who is a physician scientist role model of mine, and she's the President George H.W. Bush Chair of Cardiovascular Medicine, Director of the Division of Medical Genetics, and Vice Chair of the Department of Internal Medicine at the UT McGovern Medical School at Houston. She's received numerous honors and awards for research, including the Princess Lillian Foundation Award, American Heart Association Merit Award, and Rice Distinguished Alumni Award. And notably, she's been the director of the MD PhD program at McGovern Medical School and MD Anderson Cancer Center for over 20 years and has mentored countless trainees and junior faculty. And we're really happy to have her here today to discuss how she trailblazed a path for herself as a physician scientist while advocating for gender equity in medicine and science. And excited to hear her story. And at the end of the session, we'll have a Q&A portion. So if you have any questions, feel free to put them in the chat and Christie and I will answer them for you. Without further ado, take it away, Dr. Milewicz.
Okay. Thank you for that nice introduction, Michelle, and thank you for the invitation to talk to your program today. I'm going to go through so the sections of my talk are first just going through my training and how I made the decisions at different stages of my training in terms of which direction to take my career and how my gender kind of influenced those decisions. And then briefly go through my scientific career with some highlights and how I chose which direction to take my research. And then also some unexpected directions that I went. And then finally, at the end, I'm going to talk about what prevents women from being successful as physician scientists, and what I think my suggestions are to trainees in terms of how what you can do to increase your chances of being successful. I absolutely love what I do. My priorities are to do high quality translational research focused on a disease and to train the next generation of physician scientists. And being a physician scientist is by far the best career in the world. You get all that instant gratification of taking care of patients, but then the long-term thrill of making new discoveries, those eureka moments when you're the first person in the world to figure something out. And I just love science. I love figuring out pathways and figuring out disease processes. And one of my favorite things to do is come in on weekends and just read the literature so I can figure out, okay, we've got this piece of data. How exactly is this happening? Can I come up with a viable hypothesis to test in the lab? How do I test the hypothesis? Putting all those pieces together is just one of my major motivations, just the joy and the fun of doing science. And that's one of the reasons I'm so passionate about talking to women that are in training and, you know, going through my career path and talking about what I think I did to be that made me successful to this extent that I am. And the current data from the AANC says that although it's increasing slowly, the women that make it to full professor is nowhere near the 50% that they enter as an instructor. And now it's still about 25% actually make it to full professor. So it's getting better and better, but we're still far from parity. If you look at the medical schools over the past, 15 years, women have been entering assistant professor level at the assistant professor level at the same percentage as men, if not more. And they are just less likely to get promoted and making to full professor. So I give these talks to try to help women start thinking about what the issues are and how they can address those issues, even as students in the MD PhD program. So I'm going to go ahead and share my screen so I can show you the slides that I have. I really don't mind being interrupted during my talk. So if you have questions, put them into the chat and Michelle can interrupt me if there's something that's pertinent that could be answered immediately. If not, we'll wait until the end of the talk. So I just want to go quickly through my training and where I made decisions. I went to Rice University as an undergraduate because I really liked Rice and I wanted to stay in Texas because my family was here. When I was there, the chair of biochemistry, when I was trying to make a decision which MD PhD program to go into, the chair of biochemistry, I was a biochem major, he said, you should go to UT Southwestern and work with these investigators up there called Brown and Goldstein. They're doing really interesting stuff on cholesterol metabolism. Well, I did that. I ended up going to following his suggestion. I went to to complete my MD PhD at UT Southwestern, and I did work in conjunction with the Brown and Goldstein lab during my PhD. Brown and Goldstein, after about 6 years after I left the lab with my PhD, they won the Nobel Prize for identifying the LDL receptor and the pathways that control cholesterol in the blood. And that was the basis for the development of statins. So they made a big impact on medicine through their work. And two things I got out of that was, number one, that they use genetics to find the LDL receptor. They used a young child with very elevated cholesterol to figure out there was a specific receptor that took up LDL particles into the cell. And then second of all, it made me swear that I would never work on cholesterol metabolism ever again, or atherosclerosis, because the field was so top heavy and so competitive that I thought I just really want to go into a separate field and not where it's not so top heavy. I hadn't really faced anything in terms of gender bias until finishing up the MD PhD program. There were 4 of us that finished up at the same time, and the directors selected one of the students to really groom him to a residency at Harvard. And it wasn't until a few years later that I was thinking, why did they do that? You know, I had better grades and more publication than this man in the program, and I come back to that maybe that was a little bit of gender discrimination at that stage. I went on to do my internal medicine residency and at UT Southwestern, in part because I became involved with one of the surgical residents, and we went on to get married. So I ended up choosing to stay in Dallas because of that relationship. And the chair of cardiology at the time was Jim Willerson, and he was a fabulous physician scientist and felt very strongly about promoting women because his mother had been a physician and had been discriminated against her entire career. So he talked me into starting the cardiology fellowship, but I sort of realized about a year and a half into it that I really wanted to focus on research for my career, and I didn't want to spend another 2 to 3 more years learning how to do put in stents and other things that I knew, procedures that I knew I wouldn't be doing for the rest of my career. And I knew I wanted to focus on genetics. I had learned that from Brown and Goldstein, how powerful it was in terms of figuring out pathways and understanding disease processes in patients. And so I switched to a medical genetics fellowship. And once again, I went to Brown and Goldstein Goldstein had done his genetics fellowship at the University of Washington. So he said, this is really the place you need to go. And it is one of the top centers for medical genetics training. In addition, there was somebody there that was running a lab working on the genetics of connective tissues disorders, Dr. Peter Byers. And I was really interested in Marfan syndrome. I'd seen a patient with Marfan syndrome and had decided that I wanted to work on identifying the gene for Marfan syndrome. So Peter, when I interviewed, he said he was more than welcome. I could join his lab and work on Marfan syndrome, although he was mostly focused on collagen disorders like osteogenesis imperfecta. So I went there and started my own research on Marfan syndrome in his lab. I was very lucky. The work I was doing, I was getting biopsies from Marfan patients and looking at a particular protein called fibrilin-1, trying to figure out if Marfan cells handled that protein differently, and they definitely did. And I ended up having a plenary talk at the genetics meeting the year I was going out to look for faculty positions, and that it gave me a lot of exposure in terms of looking for physicians. And I had a large number of offers for positions. I was very lucky, including offers from Francis Collins, who was at the University of Michigan, and they had genetics there, and then also Victor McCusick at Johns Hopkins, who was sort of the grandfather of human genetics. But I really wanted to come back to Texas, and I really wanted to come back to Houston. I loved my time at Rice, and I love Houston. It's a port city, it's very diverse, and it's very easy place to live. And so I was getting all these offers, and I was, and some of the offers were, the startup package was very large, much bigger than anything I was being offered by UT Health here in Houston or by Baylor across the street. Both institutions were trying to recruit me. And so I had to negotiate to try to get them to raise their offer in Houston, because that's where myself and my husband really wanted to go. And I had no idea how to negotiate. So it turned out I would be on the phone, and my husband would be writing notes telling me what to ask for and how to do it through overhearing my communication. I think it's very difficult for women to learn those negotiation skills, to ask for what they want, or to say, listen, this program's offering me X amount of dollars, and to come to yours, I really need that offer, or something, preferably something higher. That kind of request doesn't come easily to women. And so I was very glad that I had this coach, my husband, to help me do those negotiations. I ended up, like I said, having offers from both Baylor and UT Health. At Baylor, the head of genetics at that time, the head of the department I was going to go into at that time, when he made the offer to me, he sat me down, and I was pregnant at the time with my first child. And he said, Dianna, you can have this child, but if you come and join my faculty, don't have another child. Women never make it, rarely make it when they have children, and if they have two or more children, they definitely don't make it. And the chair of the department at UT Health, and turned out to be Jim Willerson, the person that had been chair of cardiology that was fully supportive of women. And so that sort of made it very easy for me to select UT Health over Baylor, because it's really important for your goals as a physician scientist to align with the people over your division director, your chair. And when you have somebody telling you that, don't have a second child, you won't make it, that is probably not a good situation to go into. I'm hoping at this, in this day and age, that nobody has to make those types of decisions or hears that kind of advice, gets that kind of advice at this time. But don't be surprised what happens as you go along in terms of gender biases. So I ended up at UT Health, and I was, like I said, very interested in Marfan syndrome. My data had helped figure out that FBN1 was the cause of Marfan syndrome. And the reason I was interested in Marfan syndrome to begin with is patients with Marfan syndromes have formed thoracic aortic aneurysms, and they will go on and have an acute type A dissection where the blood enters the wall of the aorta and tears the wall apart. And I was just fascinated that patients with Marfan syndrome grew so tall, and at the same time, had this overgrowth of their bones, but at the same time had this very severe cardiovascular condition. And it was also about the time when they were figuring out that if they surgically repaired the aneurysm, they could prevent the dissections in patients with Marfan syndrome. And with a type A dissection, our data says that even now, 50% of the people that have a type A dissection never make it to the hospital. And another 10 to 20% die once they reach the hospital. But if you can figure out who's at risk, you can repair the aneurysm and prevent the dissection. So here was a disease, aortic dissection, where it's been mostly managed by surgeons, and there wasn't a lot of molecular understanding of the disease. And it was a wide open field, and like atherosclerosis and hypercholesterolemia. So I decided to focus in on aneurysms and dissections when I started. So I'm going to skip that and just say that when I started here, we knew by then that FBN1 mutations caused Marfan syndrome, and this is the very tall stature that these individuals have. So we've known about the FBN1 mutations for 25 years, and we still do not know why the bones grow too long. It's something that Francesco Ramirez is working on at Mount Sinai, but we still do not have a good understanding of this particular phenotype. We have a better understanding of the aortic disease, and some beautiful work by Halditz at Hopkins really showed that if you block TGF-beta signaling, if you could block aortic disease in a Marfan model. And so by like 10 years into my career, the dogma was that too much TGF-beta signaling was the cause of aortic disease and Marfan syndrome. But after it was identified that patients with Marfan syndrome had an FBN1 mutation, we realized there were people in clinic that had FBN1 mutations that looked like they had Marfan syndrome, but did not have FBN1 mutations. And my group and other groups went on to identify a number of genes, all involved in TGF-beta signaling. Once again, coming back to TGF-beta signaling as being important. And all these genes are now labeled after Halditz and Bart Loeys, and called Loeys-Dietz syndrome 1 through 4. Finding these genes has completely turned the field upside down, because these are all genes involved in protein involved in canonical TGF-beta signaling. And if you look at the signaling pathway, we're hitting every single gene along the canonical pathway. There's mutations in the genes for 2 out of the 3 TGF-beta ligands, mutations in the TGF-beta1 and 2 receptors that come together to phosphorylate SMAD3 and SMAD3 mutations. And then SMAD3 complexes with SMAD4 to move into the nucleus and change transcription. And there's mutations in SMAD4. And all these, the real paradox that emerged in the field with finding these mutations is that they're uniformly loss of function, they don't lead to gain of function. So this turned everything upside down in terms of what role TGF-beta was playing in this disease. While Hal was working on the Marfan mouse, I actually did a simple clinical study and sent a genetic counselor to get family histories from people that were admitted into the hospital for repair of an aneurysm or a dissection. And this was to figure out, focusing on the majority of patients that did not have Marfan syndrome and didn't have features of low esteets. And these, through the study, we determined that up to 20% of patients with thoracic aortic aneurysms or dissections have a first-degree relative. And we could pull out families like this, where it's like 3 generations in this pedigree of individuals with aneurysms that go on to dissect if they're not surgically repaired. And so I turned my focus on trying to find the genes in these families. And so it started a cohort of thoracic aortic disease families. So these are families that initially did not have Marfan syndrome or features or mutations in those features of low esteets, but had 2 or more members with thoracic aortic disease. And we're up to over 10,000 families and over 1,000 families, and that translates into over 10,000 participants in our study. And these families come from around the world. We really, through the studies that we continuously published on new genes, we've become very dominant. My research program's become very dominant in the field, which is a nice place to be, especially since all of this came out of just a simple clinical study. And it really shows the power of being an MD PhD. I don't think a PhD would have felt comfortable doing that type of study, whereas I felt, very comfortable sending somebody in to talk to the surgical patients and getting all the human subjects approval. And I think at some point in your career, it is good to spend some time learning how to write a consent, how to consent a patient, and how to write a human subjects protocol. And I actually learned that as a resident. I did a research rotation at the Clinical Research Center at Parkland Hospital, where they taught us how to write. We were required to write a clinical protocol and an IRB protocol, a consent and protocol as part of the rotation. So, think about making sure that this is included in some ways, because it does open the door for you to pursue research. And I wouldn't be in this position had we not been the first one to define that 20% of the families had this autosomal dominant pattern of inheritance of aneurysms and dissections. And so we figured out really early on that the clinical phenotype we were seeing in these families could vary a lot. So we hypothesized there'd be a lot of genetic heterogeneity for this condition. And it turns out we're up to like 20 genes at my lab and a few other labs worldwide that have identified. In people without Marfan syndrome, fibrillin still very commonly mutated, as are those TGF-beta receptor genes. We do have families now with SMAD2, but we have also a host set of genes that disrupt the smooth muscle cell and not TGF-beta signaling. And these all we've shown through studies in mouse models and cell studies, and even working with molecular biophysicists, that these mutations disrupt the ability of the smooth muscle cell to contract. So, we found this, the first two genes were identified, ACTA2 and MYH11. These are like the two major contractile proteins, smooth muscle alpha actin and smooth muscle myosin heavy chain, that make up the actin filament and the myosin filament that are the structural components of contraction. We found these right at the height of, everything, the hyper TGF-beta, excessive TGF-beta signaling is absolutely the primary driver of the disease. But looking at the data in terms of having mutations in the extracellular matrix and in the smooth muscles contractile complex really made me think about what may be going on with the disease. And it was already well known that if you look at the aorta, there's a structure called the elastin contractile unit, where elastic fibers, which are there's 50 layers of elastin fibers in our aorta, and between every single elastin fiber is a smooth muscle cells. And there's extensions down to that come off of the elastin fibers. And at the end of the extension are structures called microfibrils. And the major protein in the microfibrils is fibrillin 1. And there's a RGD sequence in fibrillin 1 that then links up to integrin receptors. And then within the cell, integrin receptors link up to the contractile unit. So there's like this beautiful design for the cell to sit there and sense biomechanical forces on the aorta. So we're disrupting the major structural component here, the major structural component here. And in addition, I don't want to go through all the other genes, but many of the other genes were affecting different minor components of this. And that really made me stop and think that maybe, you have this sensory structure for adaptive remodeling, when the pressures go up or go down. And that if you alter this structure, that you get the maladaptive remodeling. And I put forward this proposal that, you have these mutations that disrupt that elastic contractile unit, and it's there to maintain homeostasis across the cells. And that when you disrupt it, you upregulate pathways, and that can include angiotensin, TGF-beta, MMPs, proteoglycans, a whole lot of signaling pathways that's been shown to be to occur, and that leads to disease. And the one thing about this pathway over the excessive TGF-beta signaling is it makes sense for hypertension too. And in this case, there may not be a defect in the sensing, but instead the forces across that unit remain high in poorly controlled hypertension, and then that is the number one risk factor in people with thoracic aortic disease that don't have a mutation in one of those genes. So I actually put this out in the late 2000s as a possible alternative driver of thoracic aortic disease, and it actually wasn't referenced that much, and people didn't pay too much attention. But then, we started publishing data that those focal adhesions are activated when we made mouse models of ACTA2 and MYH11 mutant mice. And in 2014, one of the science editors read this review and some of my papers, and he came to me and said, will you write a review for Science that updates this pathway? And I did. And since then, I've been invited. This is only a short list, but I've been invited to write many reviews to update. And especially now, the entire field has essentially changed. And it was almost like turning a huge ship. It just took time and a lot of reviews and discussion, but now I think this has received a lot more attention in terms of the research and the mouse data coming out of a consortium that I run, looking for the molecular pathways, in particular those that drive dissection, really supports this pathway very strongly. And so it's been kind of nice to see this change in the focus of the field over time, but it did take a long time to change the field. Now I just want to talk briefly about 3 directions that my career took that kind of surprised me. One was what happened to this young man here. This was early on when we were first mapping, using linkage to map disease genes, and his family participated in our study. And through linkage analysis, without even finding the gene, we could tell him he was affected. And he was young at the time, and he didn't really think he was risk, despite the fact that his father, he was at risk for a dissection. And that's despite the fact that his father and his uncle and his grandfather had all had dissections. But when he was in college and one night he was studying, he started having chest pain, and he went to 3 different emergency rooms. And he walked in and said, I have a family history of dissection, and I'm having chest pain like my dad did before his dissection. And they said, no, you don't look like you have Marfan syndrome. And that's the only inherited form of, it's the only way that the disease runs in families. And all 3 emergency rooms sent him home. And a couple hours after the last ER sent him home, he died of his dissection. And then at the funeral, his brother started having chest pain. We had already told him he carried the defective gene. And the father took him into the emergency room, and they said, oh, he doesn't have Marfan syndrome. Don't worry about it. And the father's like, I'm not leaving until you image my son for a dissection, because I just buried one son. And it turns out that he was having a dissection and went to surgery, and he's still alive. So, but this was like a slap in the face. By the time that Tyler died, we had already had published a fair amount of clinical data about this being inherited in families, and we're just starting to identify genes. And it was clear that none of that was getting out in the care of the patients, and that I had to take more, if I wanted my research to impact what was going on in terms of patient care, I really needed to be active in getting it translated out there. And so about the same time of shortly after Tyler's death, I was contacted by Kevin Helliker, who was an editor at the Wall Street Journal, and he himself had an aneurysm. He had read my papers, and he was calling me to say who in the family needs to be imaged, and can we participate in your study, and so on. And I said, it would be really interesting if you could do an article on these families and how poorly managed they are, and how people don't read the literature and don't keep up to date, even the emergency room physicians that are responsible for diagnosing dissection. So I said, we can provide all the families for you to talk to, because it wasn't just Tyler that was being mismanaged, it was other families. So Kevin took this on. He wrote a 9-page article, an article that would have taken up 9 pages of the Wall Street Journal. So Thomas Burton stepped in to edit it down, but it did show up on the front page of the Wall Street Journal. And this changed things overnight. So it really confirmed that more physicians read the Wall Street Journal than the New England Journal of Medicine, in my mind. And I've got to say, Kevin Helliker and Thomas Burton went on to win the Pulitzer Prize. And the day Kevin won, I called him up and I was like, you owe me. I can get an article on the front page of the Wall Street Journal anytime I want, right? And he confirmed that. But I was glad to see that all his effort really was rewarded. And about the same, when this came out, about the same time, John Ritter, an actor, died of a dissection. So his widow, Amy Yazbek, and Tyler's mother, Deborah McMillan, helped to launch the John Ritter Foundation, and I've been actively involved in that. And this is really their goal, is to raise money for research, but also, improve the diagnosis of dissection, make sure that people know about the genetic risks, and to really help people when somebody dies of dissection, because it's not people don't commonly know about dissection, even though it has ranked as high as the 13th leading cause of death in the US in the past. So we have all these, so that's one direction. And I've been very, very involved in not only the John Ritter Foundation, but the vascular EDS, Marfan Syndrome, and a lot of foundations where vascular disease is a major feature. But we also had all these new genes for heritable thoracic aortic disease. And we've known about Marfan syndrome since the 1950s. So how to manage that disease was actually pretty well understood, but we didn't know how to manage the other diseases. And so I launched the Montalcino Aortic Consortium of aortic centers worldwide to collect clinical data on every single gene that causes thoracic aortic disease, so we can know how to better manage these families. And one thing that amazed me is how willing physicians are to take their time and enter data into a database if they see that somebody goes in and use that data to improve care. And I am just humbled by all these physicians around the world that have agreed to help with this project. And I feel thankful. And this really illustrates another thing that is good about being a physician scientist. This MAC, the Montalcino Aortic Consortium, is totally funded by the Timerty family in Canada, one of the wealthier families, and it's through the Genetic Aortic Disease Association of Canada that they provide the funding. But they offered to have the first meeting in their villa and vineyard in Montalcino, Italy. And so I named it the MAC so that we would continue to have meetings there, and we have. As it's grown, they've continued to do it, and we'll be back there in the fall. But and then they have fully supported the consortium. So it's been, I never thought I'd be doing clinical research, but this was a need, and we had found the majority of the genes, and it was the next step to be done to make sure those families are managed correctly. And I don't have time to go through all the data, but I think I'm going to skip this, but it really has implications for gene-based management for this disease, and we're planning to write those. We've been publishing papers over the years and plan to write this year the gene-based treatment guidelines, so that once a patient has a mutation in SPAD3 or ACTA2, they are followed correctly. And then finally, probably one of the biggest surprises to me is what came out of just the clinical data from the families with ACTA2 mutations. And I had used this family, we identified this family they actually contacted Debbie about who found the gene in her family. And then she said, oh, it was Dr. Milewicz in Houston. So they called up to participate in our study. And we found through, this was still back in the days where we were doing linkage and then sequencing genes under the peak, we found ACTA2 mutations. And then in this family, everybody with an ACTA2 has an orange symbol, and the black is the aortic disease. And you can see not everybody with a mutation has disease, and it's only about 50% penetrant for thoracic aortic disease. But this guy got up and said, when I was recruiting the family, he got up and we were meeting in an auditorium where they live. And I had flown to meet the family and go through the consent and everything with them. And he said, there's a lot of people with really early onset coronary artery disease and no risk factor. Could it be the same gene? And I said, no, an aneurysm, the artery gets big, coronary artery disease, it closes off. I don't think it's the same gene, but we went on to have some data that I won't go into that maybe it was the same gene. So we went back and got all the details about the coronary artery disease and also another occlusive disease, premature stroke. And now you can see if that, he had early coronary artery disease and had the mutation, and it was segregating with both diseases. This is really early onset. This guy had his first event at the age of 33 with no risk factors. So, and we went on to show in some other families like this one, we just reported out a different ACTA2 mutation to this family when this young girl at the age of 16 had a stroke and had features of Moyamoya disease. And that's where the carotid arteries are wide open until the internal carotid artery goes into the brain and they become occluded bilaterally. And this is actually her angiogram. So we figured out that ACTA2 mutations can cause coronary artery disease and also this really early onset Moyamoya disease. And I sent it to the New England Journal of Medicine, and the reviewers came back and said, there is no way this is true. And one reviewer even said I was making up the data. So, which kind of, obviously made me mad. I went on to submit it to Nature Genetics, and there the editor knew me quite well because I published there. And he said, based on the reviewer comments, I can't publish the full story, but I'll publish the thoracic aortic disease part as a full article in Nature Genetics. And then I went on to submit the rest of it to the American Journal of Human Genetics, which is kind of the next step down genetic journal. So it all got published. And after that, I contacted by families and physicians worldwide about children that had Moyamoya disease a few months after birth and aortic disease by requiring surgical repair by the time they were in their early teens. And they all have the same de novo mutation, this RJ179 in ACTA2. So I have switched my research program to figure out why mutations in this one gene can cause all these different diseases. And we're actually putting out a large number of papers on that this year. And the story is absolutely fascinating. Probably one of the most fun things I've done is figuring out how you can have one small gene, it's a relatively small gene, and cause all these diseases. So just my final thoughts as a scientist, when you start out, you really need to focus and become an expert in a narrow area. And that's the way you build up your national and international reputation, so you can, get those grants, get the promotion and so on. It's really important to learn to collaborate. Every time I find a new gene, I go through the literature and figure out who's the world's expert and call them up and say, oh, I found mutations that cause this vascular disease. And by and large, they are thrilled to collaborate because, in some cases, when we found myosin light chain kinase, the person had been working on that particular gene and protein for like 20 years. Be able to communicate. You really have to make sure your writing skills, your ability to lecture and grants, that you have those skills down. And management skills. It shocks me how much management I have to do in terms of budget. I run a multi-million-dollar research program now, and also management in terms of people skills. And I really have honed those over the years because I was not a born manager in terms of, getting the best out of people based on their own personal personality. Believe in your data and be willing to go against the dogma. I think a couple of stories I told illustrate that. Use your patients and medical observations to inform your research. That's your real niche as an MD, PhD to be successful, and be lucky and enjoy the ride. It is a lot of fun. And then I just want to talk briefly about being a woman in science. And most of you know these external forces that really hurt women, that, they request promotions at the same rate of men, but are slower to get promoted. I went to my chair to get promoted, and he said, no, wait a year. There's this man that needs to go before you. And we had started at the same time once again, and my career was just as good as his, but I got put off a year because they didn't want to put up multiple people in a particular area for promotion at the same time. Women are judged and assessed by different criteria. Women are often hired and promoted based on past accomplishment, while men are hired and promoted based on future potential. I see that over and over again. Women are not nominated for national and international awards, and all of us need to be better at doing this. When I was head of the MD PhD directors group, we actually celebrated 50 years of MSTP funding, and we wanted MSTP graduates from all levels, like those still in training, assistant up to full professor, to talk at a symposium. So I sent out an announcement to the directors to nominate people to talk, and we got about 100 nominations. There was one woman. That was it. One woman. That tells you how bad it is in terms of people thinking of women for talks and awards and so on. I'm on the AAP Council, and we put out for a major physician scientist award, got a large number of nominees. Once again, I think it's just one woman that was nominated. We have difficulty networking. I can't go to the bar to watch football with guys because, which often happens at meetings and so on. You just don't, I don't like football and I can't talk it. So it sometimes makes networking difficult. And I've had this happen to me over and over again. If you negotiate hard, they are less likely to like you. And I can give you many examples where I was called names for just negotiating for what I was should be given. And women can be overly included on committees. They really have to often have a token woman on committee. And all this comes back to unconscious bias. And I think you've probably talked about this quite a bit at some of the other talks, so I won't go through this. These are things that drive me crazy, that may be other aspects of unconscious bias, like women get interrupted at meetings more often than men. This happens to me over and over again. And I've actually stopped meetings to tell the men to quit interrupting me. It's become, at times, is so annoying. And I don't know if you remember this, but a few years back, they came out with a study saying that it happened on the Supreme Court. The women on the Supreme Court got interrupted more than the men did. And it's like, no surprise to us women at all. We realized that was happening all along. Men take credit for an idea voiced previously by a woman at meetings. That happens over again. And more often, men can say they can get something done, and women will not make those kind of pledges when they can't make sure. And there's internal forces that I think you really need to think about in yourself. I spend a lot of time in my mentees telling them they can do it. They have the skills and they have the knowledge and the ability. And it all comes from a lack of confidence and self-doubt. Modesty, I think women are way too modest. And I always ask myself, what would that guy do? Would he be modest about doing this? And that kind of helps me readjust my own modesty. I think if women say yes to too many menial tasks that do not contribute to career goals, so keep that in mind. Women are more collaborative, which often makes them look more indecisive and differential. But at the same time, because they're more collaborative, they can take leadership roles and pull people together. And I found that that is definitely true in my career. And then childcare responsibilities. Once again, there was a study out a few years ago that said for women physician scientists that they still assume the majority of childcare responsibilities. And that means you're doing extra in terms of the family, but I view it as a double-edged sword. I really, you know, when my kids woke up in the middle of the night crying because they were sick or scared, I wanted them to call for me, not for their nanny, not for their father. I wanted to be there for them and be their primary caregiver. So it's always something that sort of tears women different directions. So just a quick thing, so the things that I think I did that made a difference in my career. You've got to set your goals in terms of what you wanted. And like I said, my goals were number one, have a happy family life, but also to do substantial science and train the next generation. So I've turned down offers to look at dean position chairs to really, and have not switched my career goals. And because of that, I have always tried to reach to keep the balls in the air of those goals, of those things that lead me to those goals. So, you know, making sure the grants go out, the papers go out, but at the same time, I'm probably one of the slowest reviewers out there. My house is not decorated. My clothes tend to be, I don't go shopping that much, so they tend to look a little worn. You can't keep up with everything, so you have to pick which balls to let drop and just realize that you're doing it. It's important to surround yourself with a community that supports your goals. I talked about the chair, but also I've had a community of women that are physician scientists that have helped support me throughout my career, and that community is so important. Learn to negotiate. I've talked about that already. And finally, make sure you're looking at your peers and prepare to go into battle if you need to make sure you get that space you deserve, the promotion you deserve, and so on. And I just want to recognize all these collaborators, and in particular, all the people in my research program, but also all the sources of funding. And I continuously have grateful patients that raise money or donate money. And it's just, like I said, it's a pleasure to be a physician scientist and really impact people's life, and people can turn around and be generous with that. One of the big advantages of being a physician scientist. So I'm going to stop there. So hopefully there's a time for a few questions. I might have gone over a little bit.
Thank you so much, Dr. Milewicz, for sharing your journey, and also for all of that advice there at the end for us trainees and people still building their careers. We do have some questions in the Q&A. First, we have one from an undergrad junior pursuing biology at Spelman College. And one of the questions she asks is, as a woman, how do you manage microaggressions in a male-dominated workplace?
How do I manage microaggressions in a male-dominated workplace? I like I said, I think like interrupting people at a meeting is a form of microaggression. And, I will tolerate it to a point, and then I will say, enough, this is not appropriate. You can't continuously interrupt me every time I time to talk. And like I said, I do that at meetings, but I try to do it in a non-confrontational, and if possible, bring in some humor. But I think it's difficult, and sometimes you just walk away really frustrated, and don't have a way to address those microaggressions, and you've just got to continue to persevere and continue on what you want to do with your career and not hold a grudge.
Great advice. Yes. Difficult question.
Yeah, difficult question, but it's certainly they're there all the time. And it's just, like I said, I think most of it stems out of unconscious bias, but it just makes me so mad when I come up with an idea at a meeting and some guy follows up on it, and then all of a sudden the entire group is saying, oh, that was such a great idea, Jim. And then Jim never says, oh, by the way, it was Diana's original idea. You just, but at the same time, you can't say, wait a second, that was my idea, because you sound petty. So it really, sometimes you just have to let it go and just keep on persevering and not become hung up on it, because it, getting to where you want to be is more important than really trying to correct all those microaggressions. Hopefully over time they will go away, but you can't correct all of them.
Yeah, absolutely. And I think several times there at your talk, you talked about negotiating, times when you negotiate in your career, and then also in your advice to us to learn. And we have a question in the chat. How can we learn those negotiating skills? And especially, how can we be sure if we do negotiate that we're not considered difficult women or just, difficult people?
Okay, the unconscious bias is there. So if you go to negotiate for what you deserved, you will face that unconscious bias of being aggressive, and that's just something you have to live with. I think if you continue to be a good collaborator, somebody that is, what women do well, nurture the mentees and so on, you overcome any of that negative stuff that is generated from tough negotiation. But there's at one point where I just went to my chair and said, I've got this new grant, I need some more lab space. And he took it away from somebody that had adjoining labs that had lost all their funding. And I walked into the doctor's lounge at the hospital, and the guy was at the other end of the doctor's lounge. And mind you, I didn't ask for his labs or anything. And he just screamed out, "You bitch," at the top of his voice to me when I walked in. And, it's just amazing how inappropriate people can be, and in terms of treating women, because he would have never done that to a man, never. So in terms of negotiating skills, I think like even Biden negotiating with Putin right now, it's really interesting to see what's going on with that negotiation. And I was listening to some commentary about, you don't want to box Biden into a corner. That's not the way you do negotiations. So you can look at negotiations like that, but there's some videos that were made back in the 1960s or 70s based on a book called Getting to Yes, that goes through just simple negotiation. And that book is actually a very brief book. And so if I go into tough negotiation for something, I actually go back and watch those videos again, because it helps you a lot to refresh, what you don't want to do and what you do want to do. And then some of it is just assessing what that person needs. Like I knew Jim Willerson, the chair that when I started here, that if I had a problem, the best thing was to go in with a solution, because he was more interested in solving things quickly than actually worrying about where the resources went. So I went to him right after joining the faculty about needing a parking space closer in, because I was running home to breastfeed my child at noon. And he actually, I said, I need a parking space right at the front of the medical school, because going through the parking garages takes, an hour to get in and out. And he said, just take my parking space. And so I had like the best parking space right at the front of the medical school. But I learned early on going to him to go in with a solution, because then he could work on just making the solution work. So just also know what works for the person you're negotiating with.
Awesome. Yeah. We have another question. How would you suggest going about finding a mentor who supports gender equity at an early stage in career as a physician scientist? Actions are louder than words, but it can be harder to distinguish. So how can you really pick up on who is going to be a good mentor for you, especially, in this day and age when I think everyone would probably say they're fighting for gender equality, but who will actually stand there for you and do it.
I think that it's a good idea to look at and talk to the people that have trained under that individual, because that will give you a lot of insight into what kind of mentor they are.
I think it just hit 5 o'clock, and I know you have a lot of things you have to do. So just wanted to wrap up by thanking you so much again for making the time to talk to us over here in New York City and share your really incredible journey and progression of your career, and also all of your words of advice, taking a lot of them to heart. So thank you so much, Dr. Milewicz, again, and thank you all for attending the talk.
Yeah, thank you for inviting me.
Yeah.
Good luck with your training. Let me know if I can help you in any way. Okay.
Thanks so much, Dianna. It was great having you visit us.
Yeah, thank you.
And Christie and Michelle, thank you.
Thank you, everyone.
Bye, everybody.